Hey everyone, welcome back to chapter 25, the digestive system. This is going to be part 2. So when we left off in part 1, we were discussing the fact that when you consume food or you do your ingestion, part of what we have to do is we have to follow the GI tract from mouth all the way to anus. And part 1 basically walked us through the oral cavity, the pharynx, into the esophagus, all the way down through the thoracic cavity into the little hiatus or opening in the diaphragm, and we made our way over to the stomach. So what we're going to do in part 2 is we're going to continue our journey. Now, when we were discussing the stomach in part 1, I'd mentioned a few things to you. I said to you that the stomach doesn't really do a lot of digestion and absorption. It's really just a muscular sac that we tend to use for storage, but also because it creates the most acidic part of our body, we see that the hydrochloric acid that gives that very low pH will contribute to us breaking down our macromolecules, and it will also have part of our immune system in check in the fact that it will inhibit the bacteria or any type of pathogen that we might have ingested accidentally. What we also see is that the movement or the food into the stomach will start activating different secretions that are not only going to correlate what's happening in the stomach, but also what's happening down the GI tract. So we're thinking about our next stop, which will be our small intestines. So overall, what I want to do is before we finalize our stomach, I want to talk to you about the regulation of the gastric function. And this regulation is basically going to explain how do we get the stomach ready to receive food, how do we start activating the churning and the gastric secretion, and then how do we start pre-warning the duodenum, which is the top part of the small intestine, that chyme or food is coming down its way. And it turns out that this regulation is going to occur in 3 phases. The first phase is what we call the cephalic phase. This is basically the brain sending a signal over to the stomach that food is on its way. And it can be activated many different ways. It could be the anticipation of food, the sight of food, preparing the food, the smell of food. And for some people, including myself, just the thought of food will activate the cephalic phase, and it will start sending over messages to the stomach. And what we see happening is that through the vagus nerve, the stomach will start churning. It'll have a motility, start having peristaltic motion, and it will start initializing its gastric secretion because it's anticipating that food is on its way. The second part is going to be the gastric phase. Now, the gastric phase is going to get activated when bolus or chewed up food actually hits the stomach. And what we see happening is that the stomach knows that bolus has arrived because it will see that it starts to get stretched out to accommodate the food that's coming down the esophagus. And also, as the food is being dropped into the acidic environment, it actually tends to increase the pH because remember, the food is coming from the oral environment of the oral cavity and the esophagus. As it starts to initialize the gastric phase, what we're going to see happening here is that you will go ahead and have an increase in the movement of the stomach, and you'll also have several secretions that are going to start happening. One of them is going to be acetylcholine. The other one is going to be histamine, and then we're also going to have gastrin that will play a role in it. And histamine and gastrin, as well as with acetylcholine, their main goal is to increase how much of gastric secretion or gastric juice is being released within the stomach itself, as well as some of the enzymes that the stomach is responsible for producing. Remember that in your stomach, you have both examples of mechanical as well as your chemical digestion, because the stomach itself will have peristaltic motion, so it will have the movement and the churning. That's the mechanical aspect. And then you will also have chemical digestion because it will produce different enzymes within the gastric juice that will help the macromolecules start breaking down into their monomers. The third phase and the final phase of the gastric function is going to be all about the intestines, primarily the duodenum, which is the top part of our small intestines. And what we see happening here is that when the gastric activity starts to increase within the stomach, we see that we're going to go ahead and we're going to activate what's called the enterogastric reflex. The enterogastric reflex is initiated by the duodenum, and it's basically going to inhibit the stomach on a temporary basis. Which sounds a little weird. Why do we want to shut down our stomach? We're in the middle of kind of trying to process our food and getting our absorption, our digestion and absorption started. So why would we want to stop the stomach from doing what it's doing? Well, it turns out that the enterogastric reflex is basically an opportunity for the duodenum to make sure that it clears any food that's sitting in the small intestines. Now remember, at this point, the food is no longer called bolus. We're going to call it chyme, because once it reaches the stomach and it becomes the soupy mixture, we call it chyme. So if there's any chyme sitting in the duodenum or anywhere else in the small intestines, in the intestinal phase, the job will become to clear it out and pass it down into the large intestines. And in order to do that, the duodenum will go ahead and activate the enterogastric reflex so they can get rid of the old before the new chyme makes its way down from the stomach into the duodenum. There are several secretions that will help you with this role. So if you go ahead and you take a look at the bottom of your PowerPoint, you're going to see that it says the chyme will stimulate the duodenum cells to secrete secretin, CCK, and gastric inhibitory peptide. All three of those secretions are going to suppress your gastric secretion and motility. So it's going to inhibit the stomach on just a temporary basis, just a little while to clear out the duodenum, and then the stomach will be up, back and running, and your small intestines will be ready to receive new food coming down its way. We also see that as the food becomes bolus, becomes chyme, and it moves from the oral cavity into the pharynx, into the esophagus, into the stomach, we see that we have several accessory organs that will come into way, that will allow for secretions to enter, that will increase the amount of absorption and nutrient that we can extract from our food. Some of these accessory organs are going to be your liver, your gallbladder, and your pancreas. So what I want to do is I want to take a few seconds and I want to go over these three items, and let's talk about what they actually produce and how it's actually combined or it enters into your GI tract. So let's take a look at the liver. The liver sits right below the diaphragm on the other side of the stomach. It's about 4 pounds. It has 4 lobes, a right, a left, a quadrate, and a caudate. And for the liver, what we see happening is that its most important function when it comes to our digestive system is the fact that it's going to be producing and secreting bile. And to help with that function, we're going to see that we're also going to rely on the gallbladder. As you can see, I've highlighted the gallbladder for you on the illustration at the bottom right-hand side. It is a little sac that sits embedded within the liver itself. And what the gallbladder will be doing is it will be storing some of the bile for later use. Okay, so on this PowerPoint, it just kind of reiterates what I was chit-chatting with you before. It says the liver will produce the bile, and then what we see happening is that the liver will store some of its bile in its gallbladder. And the reason it does this is because if you ever sit down and you have a high fatty meal and the bile from the liver is not enough, you have an extra backup in the gallbladder that can be released. Now, I kind of went a little ahead of myself because I was talking about eating fatty foods and the bile. What I should have said to you before is the reason why we need bile in our digestive system. And the reason for that is because bile is what we call an emulsifying agent. An emulsifying agent is going to allow two solutions that naturally don't mix to mix together. And in this case, I'm talking about lipids and water. Our digestive system, when it's doing its digestion, it's also doing what we like to call hydrolysis, meaning that it uses water to lyse or to pull apart these macromolecules into their smaller subunits. And that works great for carbs and for proteins. But when it comes to lipids, when it comes to fats, Those are hydrophobic. So how can we break down the fats that we consume? Well, that is where the bile is going to come in. Bile is going to be our emulsifying agent, and it's basically going to force the interaction between the water and the lipids so that we can extract the nutrient and the energy out of that food source as well. Within the bile itself, we see that it contains many different things. It's one of the ways that your body can utilize the cholesterol that it has inside of the body. Some bile acids are in there. We also know that it has some phospholipids, different minerals, and it also does have some pigments, some coloring to it. Some of those colors are the reasons why bile itself has a yellowish tone to it. It comes primarily from bilirubin. That name sounds familiar because we discussed it when we talked about red blood cells and what happens when they die off, and we have to recycle their components, including hemoglobin pigments. And what we see happening is as the bilirubin moves down into the large intestines as part of the bile secretion, it will actually be converted to urobilinogen, which is one of the reasons why our fecal matter is actually brown. If we didn't have this coloring, our fecal extraction would be more of a grayish off-white coloring. As you can see, I'm a little obsessed with colors, that I know what color your feces would be without bile. Either way, so at the bottom of your PowerPoint, it reiterates the fact that the bile is needed to emulsify the fat, and that is going to help us with digesting it and breaking it down into its monomers. We're going to talk more about bile in a few slides when we take a look at the different food groups and the enzymes that are paired up together to maximize our nutritional intake. Alrighty, here is our pancreas. For the pancreas, we've talked about the pancreas before. So the pancreas sits a little bit behind the stomach, and what we see happening, it's an example of a mixed gland. We talked about this when we were talking about endocrine system, the system that produces our hormones. And when we did the pancreas, we talked about the fact that one of the main hormones it produces is insulin, which allows the uptake of our glucose. So pancreas is also an exocrine gland, and that's because it's going to be responsible for a lot of the digestive enzymes that we require for our digestive system. So on your PowerPoint, it says the fact that it's able to secrete insulin and glycogen, those are the hormones, that's going to be the endocrine aspect. And the fact that it's going to be able to produce pancreatic juice that is going to be filled with digestive enzymes, that's going to be the exocrine aspect. And the cells that are going to be doing the exocrine aspect, those are going to be called acinar cells. And the wide majority of pancreatic cells will be acinar cells. So they will be responsible for the pancreatic juice secretion. Here we have a little illustration on the left-hand side with the acinar cells. And as you can see, they're able to secrete a wide variety of different enzymes. Now, a lot of the enzymes they'll produce will be in what we like to call proenzyme form, meaning that they are not activated until they actually are released by the pancreas. And the main reason for that is because a lot of these are proteases. Proteases are enzymes that break down proteins, and your pancreas is mostly made out of proteins. So it doesn't want to be secreting enzymes that can actually break down the gland by itself. So what it will do is it will secrete these enzymes in its preliminary or its pro state, and only when the enzyme is released into the small intestines will the enzyme will become activated so it can break down the proteins in the food that we've consumed. Here are a few of the enzymes that the pancreatic acinar cells will be producing: trypsinogen, chymotrypsinogen, procarbopeptidase. Now notice how within the names I've highlighted in blue trypsin or peptidase, and that's because those will be the final version of the enzymes that we're going to take a look at, trypsin and peptidase. The pancreas will also give us another way for us to get amylase. You might remember your amylase because you had that secreted in your saliva. It's the one that can help you break down your carbohydrates. Your lipase, You also had lipase from the saliva. That's the enzyme that, even though it's in your saliva, doesn't become active until it gets into your stomach. And it will also give us enzymes for RNA and DNA. So those are your ribonucleases and the deoxyribonucleases. And at the bottom of the slide, I wrote down for you, why is it necessary to release more digestive enzymes from the pancreas? We've already had a bunch of them in our saliva and in our stomach. And the main reason for that is it's all about the pH. Remember that your stomach is very acidic. So if you have enzymes that are active in an acidic environment, they work perfect in the stomach. But the majority of the enzymes that your body produces are optimal at a neutral environment. So when the chyme comes out of the stomach and it's neutralized, what we see happening is that it needs a second dose of enzymes that will thrive in the neutral environment and that will continue to absorb and digest our food. And the pancreas is the perfect way for us to reintroduce these enzymes into the mix. Alrighty, so here on this illustration, it shows you how the secretions from the liver, the gallbladder, and the pancreas are dumped into the duodenum, which is the top part of the small intestines. One thing that I want to reiterate that I think it's written in your PowerPoints, but I forgot to kind of verbally say it, is when it comes to bile, it's very important to remember that bile is produced by the liver. It is stored in the gallbladder, and that both the liver and the gallbladder are able to release bile into the small intestines. And the reason I want to reiterate that is because many of us know someone who had their gallbladder removed. And there can be many reasons why you do have to go in. You might have some gallstones, some people have inflammation, and they have to take the gallbladder out. Well, what this then means is that you no longer have your storage unit for in case of those high fatty meals, but you're still able to process fat because the gallbladder was simply storing the bile. The liver is the one that is actually producing it, and the liver can also release the bile into the system. So very important to point out that the liver is able to produce, the gallbladder will store, and both the liver and the gallbladder will release the bile into the small intestines. All right, let's go back to our picture. So go ahead and take a look at the right-hand side, and you can see the liver right there. You can see the gallbladder in green. In the khaki formation, you can see the pancreas, and then the pink tubing that's towards the bottom, that is going to be your duodenum. That is the top part of your small intestines. And what this picture is trying to show you are the different ducts that the secretions will use to enter into your small intestines. So I've highlighted the most important ones. So you can see right there on your right-hand side in your first red box, you have the right and left hepatic ducts of the liver. And what we see happening is that within the liver itself, the individual cells, the hepatocytes, will be producing the bile, and they'll start secreting it, and it will be collected into the right and left hepatic ducts. These, the left and the right, will then merge together to come by what we call the common hepatic duct. And the common hepatic duct will actually be joined by the cystic duct. And when it joins by the cystic duct, then we call it the bile duct. The cystic duct comes from the gallbladder. So let's do this again. So the individual cells of the liver will go ahead and release their bile into the right and left hepatic ducts. The right and left hepatic ducts will merge together into the common hepatic duct. The cystic duct from the gallbladder will join together with the common hepatic duct to give rise to just the bile duct. So see, because both of those are able to secrete the bile. And now what we see happening is from the pancreas, we also have a duct because the pancreas is releasing its pancreatic juices with all the different digestive enzymes. And we see that the duct of the pancreas and the bile duct, that's coming from the liver and the gallbladder will combine together into the hepatopancreatic ampule, and this will then be released within the sphincter, which is the opening that guards the entrance into the duodenum, the small intestines. So the secretion of the liver, the gallbladder, and the pancreas will eventually all merge together into the hepatopancreatic ampule, which will then allow it to be released into the duodenum, which is the top part of the small intestines. I also want to just quickly highlight some of the hormonal secretions that we've seen along the way, especially when we're looking at our regulation of gastric secretion at the beginning of the PowerPoint. So here's just another quick mention of your CCK, your secretin, and your gastrin. All three of these are being released by the duodenum, and all of them will be activated by the presence of food being found within the stomach, or the stomach starting to release some of its chyme within the duodenum. And the main goal of it is going to be all to start contraction within the gallbladder and the liver so that they can start secreting the bile and the pancreatic juices. We also see that with the secretins, for instance, it will go ahead and it will secrete more bicarbonate. Bicarbonate is a perfect neutralizing agent. It will help the acidic chyme coming from the stomach to start being neutralized so that it doesn't aggravate the small intestines as it's entering the neutral environment. Alrighty, let's take a look at the star of the digestive system, and that is going to be our small intestines. The reason I said this is the star is because this is where you will have mostly all of your chemical digestion and absorption occur. Now remember, digestion means that you're able to successfully break it down into smaller molecules, so you're going to go from macromolecule to the monomers, and absorption means that we can take the nutrients and we can actually allow them to be dumped into the bloodstream so it can circulate outside of the digestive system to all the other cells that we have that have nutritional demands. It turns out that our small intestines is composed or is broken down into three sections. You have your duodenum, the jejunum, and the ileum. And those are the ones in order from top to bottom. A quick way to remember it is don't jump in. So the duodenum, some people like to say duodenum, and actually both are correct in saying. So the duodenum, the jejunum, and the ileum. For your exam, you will be responsible for telling me what's the difference between these three sections. So I definitely want to go ahead and go over to our next PowerPoint so we can kind of take a look at what makes each section special. All right, so please know the order: duodenum, jejunum, and ileum. You don't really have to worry about the length of each section because remember, our small intestines are tightly coiled by that mesentery, that see-through connective tissue that's full of the blood vessels. So you don't have to worry about the measurements. I mean, you can see I've posted them on the PowerPoint. 10 inches for the duodenum, about 8 feet for the jejunum, 12 feet for the ileum. My main concern is knowing what each section contributes. So for the first part, the duodenum, I would love for you to know that its main purpose is to neutralize the stomach acid that is coming from your pyloric sphincter. Remember, the pyloric sphincter is the narrow end of the stomach. That is where the acidic chyme will leave the stomach and enter into the neutral environment of the small intestine. And what we need to do is we need to neutralize the stomach acid by introducing it to lots and lots of mucus and bicarbonate. And this is going to be courtesy of the Brunner glands. So the Brunner glands are going to help us neutralize the stomach acid, and they're found within the duodenum. In addition to neutralization, also keep in mind that your pancreatic enzymes as well as your bile secretion are also being released in the duodenum, courtesy of the liver, the gallbladder, and the pancreas. The second part of your small intestines, the jejunum, that one is going to be known for being the place where most of your digestion and nutrient absorption is going to occur. So that is really where all the magic happens for your digestive system. And the third part, the ileum, that one's going to be known for two things. The first one is the fact that it has Peyer's patches, which we discussed in our immunology chapter. Peyer's patches are going to be basically nodules that are going to be filled with white blood cells. So they're going to play a role in our immune system to catch any type of pathogens that might still be available in the chyme moving down our GI tract. And the second thing for the ileum is that this is the end part of the small intestine, and this is where your small intestine ends, and in essence your large intestine start. And what we see happening is that the meeting point between the small and the large is going to be called the ileocecal junction. Ileo hinting towards the ileum, and cecal will be for the cecum, which will be the first pouch that we encounter when we get to the large intestine. So your ileocecal junction is found in the ileum. So just to reiterate, your duodenum is the first part. This is the part that's exposed to the pyloric sphincter of the stomach, and its main purpose for the duodenum is to neutralize the acidic chyme, and it's going to do so by Brunner glands secreting lots of mucus and bicarbonate. It's also the receiving point of your pancreatic juices and your bile secretion. The jejunum is the middle part. This is where most of your digestion and nutrient absorption occur. And then the ileum is going to be the end part. This is where you're going to have your Peyer's patches, which are your white blood cells nodules. And you'll also have the meeting point with the large intestine, which is going to be called the ileocecal junction. Within the small intestines as well, we really do see the utilization of the circular and longitudinal muscles that make up the layers of the GI tract. And what we see happening in the small intestines is that the circular muscles will do a lot of segmentation. And segmentation basically means that it's going to manipulate the lumen at different areas to create small little squares down the long tube of the small intestines. And the purpose of segmentation is basically to increase the amount of mixing and churning that the chyme does as it's being mixed with the pancreatic juices and the bile. So it really does increase the exposure of the food to the different digestive enzymes. So as you can see on the picture right there, they've shown you three little pieces of the small intestines, and everything is broken down into small little segments, allowing the mixing of the food and the digestive enzymes. At the same time that you're doing segmentation, you also have to keep the food moving down the small intestines through the duodenum, the jejunum, and the ileum. So if you see that the food is moving downwards, then what we're looking at is going to be the peristaltic motion that you've heard me say for quite a few times now already. So your peristaltic motion is going to be more of a wave-like motion. It is a coordinated motor complex that allows the food to move down the intestines. And obviously, as it's moving down the intestines, its ultimate goal is to reach the colon or the large intestines. And also, it's going to start being able to release its small micronutrient into the bloodstream as it's being broken down or digested in the jejunum. Alrighty, let's take a quick little break from moving down our GI tract, and let's concentrate on the different enzymes that I've been chit-chatting about. And my goal in the next few slides is to kind of put them all together, because on your exam, I would love for you to be able to tell me which enzyme is going to be for which macromolecule and what part of the GI tract they'll be activated in. So let's go ahead and let's start off with our carbohydrates. So our carbohydrates are basically our starchy foods. So these are things like when you're consuming rice, potato, pasta, and your body has many different versions of carbohydrates that it gets exposed to. So for instance, we have our starchy foods, as I mentioned. We have our cellulose, which we come across in the plant cell walls of any of the vegetables and fruits that we consume. We have chitin, which you find, for instance, in the cell wall of fungi. So if you guys like to eat mushrooms, you're consuming chitin. Glycogen is a food storage molecule. It's basically different sugar molecules linked together. And the goal of your carbohydrates when they're introduced into your digestive system is to be broken down into their individual monomers, so they're building units. And the monomers for a carbohydrate is a monosaccharide, a simple sugar, and the one that your body is really interested in is glucose. Glucose is your blood sugar. So anytime you have a carb, your body wants to break it down into glucose, and that glucose will ultimately go to your individual cells. Well, it will serve many different functions, including aerobic respiration. Now, what are the enzymes that our digestive system can use to help us break down our carbohydrates? Well, there's two main ones. We have our salivary amylase, because it's found in the saliva in our mouth, and we have our pancreatic amylase, which comes from the pancreas and is then dropped into the duodenum, the top part of the small intestines. Now, go ahead and take a look at the little diagram that's on the right-hand side, and I put two really big red stars next to the salivary amylase and the pancreatic amylase so that you know the name of the enzymes. And then I've also highlighted the fact that they are active in the mouth and in the small intestines. So that is your hint to indicate that it's both neutral environment. And then the question then is, well, what happens in the stomach? Because you can see the stomach is highlighted in my diagram. Well, nothing. Because it turns out the salivary amylase in your saliva, when you start chewing and the food starts mixing with the salivary amylase, it starts breaking down the carbs, and you can see the different carbs labeled: glycogen, starch, disaccharide, cellulose. You can see them labeled. It's going to start breaking down your glycogen and your starch primarily into oligosaccharides, which are a little bit smaller than a carb, but still not your monosaccharide. But the minute all that bolus hits the stomach and it's exposed to an acidic environment, the salivary amylase becomes denatured, so it can no longer be active, which means that you don't have any carbohydrate digestion occurring in your stomach because all your enzymes for the carbs are denatured. It's only when that acidic chyme now leaves the stomach and goes into the duodenum that it's then exposed again to pancreatic amylase. Thank you, pancreas, that the digestion of the carbohydrates will continue, and you can see that your glycogen, your oligosaccharide, any starch that you had left will be continued to be broken down into disaccharides, and from there on it will be broken down into our monosaccharide, which is our glucose that we're looking for. Once it's in the monosaccharide form or in any monomer form, we see that within the small intestines, in the jejunum primarily, they're able to be absorbed through brush border enzymes, which means that these enzymes can pick up the nutrients in the lumen of your small intestines, and basically to active transport or facilitated diffusion, they can dump these nutrients into the bloodstream outside of the GI tract so it can circulate throughout the body. An interesting fact that I also want to point out is go ahead and look all the way to the right-hand side of your chart, and you could see cellulose right there. Cellulose is the carb that we find in plant cell walls, and it turns out that it will pass through unbroken. Look at that. So you start off with cellulose, it passes through the mouth, the stomach, and the small intestines, and it's still cellulose. So what's happening over there? Well, we're going to have to wait a few more seconds to describe how we're going to get the nutrient out of those plant cell walls, okay? So hang in tight. I'm going to tell you guys about that in a few slides. Our next section of macromolecules are going to be our proteins. Our proteins, we want to break them down into amino acids. Most of us get our proteins from consuming any type of meat products. Obviously, you can get it from a wide variety of items, but meats and nuts tend to be the highest category to consume proteins in your diet. Now, our two enzymes for the proteins are going to be pepsin and trypsin. Pepsin is primarily activated in the stomach by the hydrochloric acid, so it is an acidic enzyme. And trypsin is going to be one of the enzymes that your pancreas releases into the small intestines, so it thrives in a neutral environment. So this is really important to highlight. So you can see on your chart, you start off with your protein, you pass through the mouth. There's no enzyme in your saliva that helps you with protein digestion. So you're doing nothing but mechanical breakdown of the protein, ripping it apart with your teeth and your tongue. You won't do any chemical breakdown until you get into the stomach and you're exposed to pepsin. And when the acidic chyme leaves the stomach and goes into the small intestines, then we see that the pepsin becomes denatured because it likes an acidic environment. So at that point, we then rely on trypsin to take over, and trypsin will allow the protein to be fully broken down into their amino acids. So please make a notation for yourself. Both pepsin and trypsin break down proteins, but pepsin is in the acidic environment and trypsin is in a neutral environment. Our last category is going to be lipids. Lipids are our fats. Now, quick reminder to yourself, lipids are hydrophobic, so remember they also need bile. Bile is going to be our emulsifying agent. It's going to allow the water in our GI tract to mix together with the enzymes and the lipids. And our main goal is to break down the dietary fats into fatty acids and monoglycerides. The enzymes that we're going to use in conjunction with the bile will be salivary lipase and pancreatic lipase. Now, salivary lipase, as you see from the name, and hopefully as you remember from part 1, comes in your saliva. However, it's not activated until it gets into your stomach because it requires an acidic environment. Your pancreatic lipase comes courtesy of the pancreas and is active in a neutral environment, so that will go ahead and be introduced into the duodenum, the top part of your small intestines. In case you were wondering, can you have lipid digestion if you don't have bile? The answer is yes, but it will be very minimal amount of lipid digestion that will occur. So your best result is if you mix it not only with the enzyme, but also with the emulsifying agent due to the fact that it's a hydrophobic molecule. A few notations, your smaller molecules like your vitamins and your minerals, those usually are able to be absorbed unchanged. So on your PowerPoint, I kind of wrote down for you your vitamins like A, D, E, and K. They basically move through with the other lipids. We discussed the fact that B12, the one that you need for your DNA replication and red blood cell production, they have to be bound with intrinsic factor, which comes courtesy of the stomach. Your minerals will tend to piggyback off different electrolytes, or they might use co-transporters. So for instance, your sodium loves to move along with sugars and amino acids. Your chloride tends to go ahead and combine with bicarbonate to assist with the neutralization of the stomach acid. And iron and calcium, your body will utilize what it needs, and if there's any excess, it will try to store a little bit, but most of the time it's flushed out of the system. Towards the top of the PowerPoint, it says the nucleases hydrolyze DNA and RNA. So anytime you eat animal or plant products, you're consuming cells, and inside of those cells are DNA and RNA. And what your body loves to do is it loves to break down those molecules so it can use the A, the T, the G, and the C, adenine, thymine, guanine, cytosine, and uracil. So the nucleotides that make up DNA and RNA, your body loves to recycle those, and it will do so by using nucleases, which primarily come from the pancreatic enzymes. We also have to kind of just quickly mention water balance. Water is extremely important to the body. Our body is mostly composed of that H2O molecule, and what we see happening is that your digestive tract on average can get over 9 liters of water a day. So part of what we see happening is that the small intestine will mostly be the one doing the absorbing, and then the large intestines will also play a small role in it. And we see that a lot of it will be controlled by the different salts and nutrients that are introduced into our GI tract. So, for instance, we can talk about a clinical application of what happens if too much water gets released or not enough water gets released from the food that we're absorbing. So at the bottom of the PowerPoint, it says that if you have diarrhea, that usually occurs when there's too little water that's absorbed by your GI tract. So what ends up happening is all that excessive water stays within the small and the large intestines, and eventually it's then flushed out through the anus. And this can happen many different ways. Most of the time it could be because we ate something that upset our GI tract and our body's just trying to flush it out as quickly as possible. People who are lactose intolerant might have an issue with this. So for instance, if you do not have the lactase enzyme, the enzyme that produces that helps us break down the sugar in milk products, what we see happening is if you decide to have yourself a big old bowl of ice cream, you're going to notice that your stomach and your small intestines start to chur a little bit. And that's because since your body can't break down that milk sugar, it's going to flush it out of the system as quickly as it can. So it's going to increase the peristaltic motion of your small and large intestines, and the water will not have enough time to kind of be absorbed into your individual cells. It will be flushed out with your fecal matter at the same way. So that's good old-fashioned diarrhea for you. The opposite is true is the food moves too slow. If the food is moving too slow through our GI tract, then too much water is absorbed, and therefore it hardens the fecal matter, and that is then where constipation will step in. So your water balance is very important to regulate your GI tract. Alrighty, so let's go back to our large intestines. So last we spoke about our GI tract, we talked about the fact that the food had gone from the stomach into the duodenum, the jejunum, and the ileum, right, into our small intestines. And you can see on your right-hand picture in the purple and the pinkish color, you can see your small intestines nicely coiled up. And from the small intestines, our next stop will be our large intestines. The large intestines in humans, I always like to describe as a picture frame. It will literally frame the small intestines. It will start at the ileocecal valve. Go ahead and find that little arrow that I've indicated on the left illustration for you. The ileocecal valve will allow the chyme to come into the cecum, which is a little pouch that sits right at the beginning of our large intestine or our colon. And from the cecum, we see that the large intestine goes upward in the ascending colon. Then it goes across in the transverse colon. It will then go down again in the descending colon. And finally, as it moves in between the hip bones, it forms the S colon or the sigmoid colon. Now, if you were to compare your small versus your large, from an anatomy standpoint, we see that the small intestine is longer. However, it has a smaller diameter, whereas the large intestines is shorter, but it has a larger diameter. It has a larger width to its lumen. So as you can see right here, it says it's only about 5 feet long, 2.5 inches in diameters. It starts with the cecum. Oh, and I forgot to mention, your little appendix also dangles off the little cecum right over there. Your appendix is, for a lot of papers will tell you that the appendix is what we call a vestigial structure, so something that we have in our body that we don't really need, which is sort of true, but there's also been a lot of new research showing that your appendix actually houses some of the bacteria that your gut uses on a day-to-day basis, and your appendix will kind of house kind of like a little reserve of that bacteria in case you ever need to replenish it. And we also know that it tends to house white blood cells as kind of like a little backup immune system in there. So it does have a function. Obviously, there's also lots of evidence that if you have to have your appendix removed, that you can continue to live a very healthy quality life. So Just a little FYI. Also, to mention that it's ascending, transverse, and descending when you talk about your large intestine, and that the sigmoid colon is what leads you into the pelvis, and it will finalize or it will end in what we call the rectum. The rectum is the straight section where your fecal matter will basically sit and wait until it can pass through the anal canal, out through the anus, and be secreted out of the body. Now, inside of our large intestines, we're also going to see that we have lots and lots of bacteria flora. There are more bacterial cells in your large intestines than you actually have cells that make up your entire body. So we have millions upon millions of bacteria cells just sitting in our large intestines. Now, some of you might be grossed out right there, but it turns out it is what we like to call a mutual symbiotic relationship. Both of us benefit from this relationship. The bacteria obviously get a housing and they get to be exposed to the food that we're eating. And we, on the other hand, get to go ahead and increase the nutrients that we can get out of our individual food products, including all those plant products that we've been consuming that have been hidden or been protected by their cellulose cell wall. Now, we talked a few minutes ago about how your enzymes, your salivary amylase and your pancreatic amylase, are not able to break through cellulose when it comes to the carbs, while the bacteria that sits in your large intestines, they can break down through the cellulose. And as they break down through the cellulose, they release all the nutrients that's inside of those plant products like our fruits and our vegetables, so we can extract those nutrients out of it. We also see that the bacteria, because it's digesting some of our food, will produce different chemicals, and part of that will contribute to the smell when we release flatus or gas. Now, when you release gas, so when you fart, it's mostly inhaled air that went into the esophagus instead of the trachea, so your body's just kind of letting it out. The reason that air has a certain smell to it most of the time is due to the production of hydrogen sulfide, indole, and scatol, which are mostly byproducts from the bacterial digestive system. All right, so your large intestines is shorter, it has a larger lumen, and in case you're wondering what exactly does my large intestines do, well, its primarily function is going to be the packaging of your fecal material. So any of your undigested material, this is the end stop for them. We're going to package them up. It will also contribute with a little bit of water reabsorption, which we talked about, but its main goal will be to pack up our fecal matter. Your feces is a combination of items that were not able to be digested or broken down. It's also things like some of the waste products, maybe died of bacteria, a little bit of mucus, some fibers, some cells that came through the GI tract. Everything will kind of package up together. Usually the contractions that will allow the feces to move through your large intestines are a little bit stronger, so we call them haustral contractions, and usually you will have mass movements, which basically means it's the compiling of the feces and getting it to actually exit out of the body about 1 to 3 times a day. So mass movement is basically the sensation you feel when you know it's time to go to the bathroom because you have to empty out your rectum. Your mass movement is triggered by gastro and duodenum reflexes, so the stomach and your small intestines will go ahead and signal to your large intestines that new food is coming down, and it's in essence time to clear out any fecal matter that might be stored in the rectum. This is one of the main reasons why usually after you have, your regular meal like your breakfast, your lunch or dinner, within about a half hour to an hour of time, you'll have the sensation that you have to go to the bathroom to pass some of your feces along, and that is then your mass movement, and that's because your stomach and your small intestines kind of gave the large intestines a heads up that more food is coming its way, so clear out any of the feces that you're storing in your rectum pouch. All right, so here is a look at our rectum, and really this is more a look at the anal canal, I should say. You can see the anal canal is the last 3 centimeters of your GI tract. You can see it has little longitudinal ridges that will allow us to secrete lots of mucus in there. Mucus will help lubricate the feces, so it makes it easier to pass it through the anus, the opening of your GI tract. We also have lots of blood vessels, primarily veins, and if those veins become distended, so they move out of their original spot, that is then how you can get hemorrhoids in that particular area. Also notice how you have this muscle called the levator ani muscle. That muscle will contract, which will lift the rectum slightly up, and it will open up the anus so that the fecal matter can kind of fall out whenever you have a mass movement. Guarding the entrance of the anus is going to be a sphincter. So if you look at the bottom right-hand side of the illustration, I placed a little red box around the internal anal sphincter and the external anal sphincter. You have 2 of them. The one that's internal is composed of smooth muscle, which means it's under involuntary control, so you can't consciously control the opening or the closing of it. And the external anal sphincter, that one's made out of skeletal muscle, so that one's under voluntary control. And part of, let me see how to say this, part of if you have to potty train a child is that you teach them to control that external anal sphincter, so to control that skeletal muscle. So if you're ever in a situation where you have to pass fecal matter, but you can't do it, that you can hold it until later. Obviously, your internal anal sphincter you can't hold until later because it's under involuntary control. But if you've ever found yourself in a situation where maybe you're in class and you feel that mass movement coming and you feel yourself kind of squeezing to hold it in, that's your external anal sphincter that you're keeping closed. And fun fact, your urinary system has the same thing when it comes to the excretion of the urine. It has an internal and an external sphincter. Smooth muscle on the inside, skeletal muscle on the outside external sphincter of it. And then if you look on top, I put a little box around the structure called a rectal valve, and I wrote down what's the purpose of the rectal valve, right? So you can see the rectal valve basically extends inwards into the rectum, and it kind of creates like a little half wall. So why do we need that? I think it's a very cool function. And in order for us to explore the purpose, we need to click over to the next slide. So let me go ahead and do that. Alrighty, so take a look at that illustration on the left-hand side, and you can see they went ahead and they drew the colon for us, the rectum, and now they went ahead and included fecal matter. Fantastic. Now find your little rectal valve. It's not labeled, but you can see it's right over there by the stretch receptors. You can see how it's like inserting itself into the rectum. And the purpose of the rectal valve is that it will actually hook and hold onto your fecal matter so that you can, get ready for this, you can pass gas without pooping. How cool is that? Because it will hold onto the fecal matter. I'm sorry, I just think it's one of the coolest things that your body does. So the rectal valve will hold on to your feces so that if you ever have to pass gas but you can't poop, you can do so. You can go ahead and relieve that pressure out of your system. All right, now this last picture right here also kind of reiterates the fact that if it's time for defecation or releasing of the fecal matter, we do have some neural control to it, and part of that will go ahead and control the relaxation of your internal anal sphincter, because remember that one's smooth muscle under involuntary control. And then your last step will be to activate the relaxation of your external sphincter, which is done by your skeletal muscle and is under voluntary control. Alrighty guys, that is our last slide for our digestive system. Let's meet up in the discussion sessions and let's see what's going on. Alright, we'll talk soon. Thank you.